The YouTube live. Yes, we have done it. Good.
Dr. Ricardo is there.
Yes, we can start. Jayaprakash.
Okay.
Just a minute. Professor Jayaprakash.
Yes, yeah. A very good morning to all. So it is my pleasure to introduce the first speaker in this session two. There are three speakers. The first session will be done by Dr. Sophie Funk. So it's my pleasure to introduce her. So Dr. Sophie Funk, she has a background in biology and ecotoxicology. She did a PhD thesis on how to improve the ecosystem resilience to reduce the environmental impact of oil spills. And at Plastic Soup Foundation, she coordinates various research projects and provides scientific support for the health campaign. So with this, I would like to invite Dr. Sophie Funk to present her talk. Yeah, thank you. Please go ahead, Sophie.
Thank you so much for this introduction. Let me not forget share sound. Excuse me. Yes. Does this work like this?
Yes.
Okay, perfect. Well, good morning, everybody. Thank you so much for having me. It's a real pleasure to speak at this virtual conference. I was introduced, but I'd like to introduce myself quickly. My name is Sophie Funk. I have a background in academia, working as a biologist. And now for 2 years, about 2 years, I've been working as a researcher at Plastic Soup Foundation, an Amsterdam-based NGO. And I'd like to talk to you today about microplastics and the effects on human health. So what is it that we do know already, and what are the things that we don't know already? But before I go into this, I would like to briefly discuss our organization, Plastic Soup Foundation. I'm not sure if everything can be seen in my slides, but this organization was founded by Maria Westerbos, our director, in 2011. And from a very small organization, in less than 10 years' time, we grew into one of the largest and most important advocacy groups tackling plastic pollution. And we have a very simple but clear slogan, which is no plastic in our water and no plastic in our bodies. And we try to achieve this goal via various campaigns that we run. For example, Beat the Microbeats. With this campaign, we aim to ban all microplastics from personal care products. And a different way to reduce emissions of microplastics to environment is via our Ocean Clean Wash campaign. And with this campaign, we aim to reduce synthetic fiber emissions via clothing by 80% in the coming years. And of course, also we focus on macroplastics. Last year we organized World Cleanup Day, and we'll do this again this year. So a lot of people go onto the streets and collect waste. So this way we are cleaning up the streets, but we're also actually collecting very valuable information, because we ask everyone to include the information about origin of the plastic and brand of the plastic in an application that we developed. And then a project that we do in collaboration with 2 NGOs in the Netherlands is Clean Rivers. And with this project, our ultimate goal is to stop any plastic from reaching our rivers. Also, we find it very important to educate the next generation on this issue. And for this reason, we developed educational material for both primary and secondary schools. And then our latest campaign is our health campaign. And that's what I want to speak about today. In December 2018, we created a research and advocacy alliance called the Plastic Health Coalition. And in this coalition, various national and international research and advocacy groups have joined. And we've joined forces to enhance research into the health effects of plastics on human health. But apart from this, we also want to make sure that all this research is disseminated to a greater and wider public to make sure that there's more awareness around this issue. And in the end, we want to be able to answer the question, does plastic make us sick? So why did we feel this was necessary to create this coalition? Well, microplastics are all around us. That's the issue, in our opinion. Microplastics have been found on top of the Himalayas, in the deepest ocean trenches, on the North Pole, South Pole, but also much closer to home. It's in our food, it's in our water, it's in the air of our indoor homes. So this means that we eat, drink, and breathe plastics every day. And interestingly, the effects of microplastics have been actually quite extensively studied for marine organisms, but strangely enough, it's hardly been studied for humans. So I'd like to go into some ways how microplastics may affect our health. And one of the things that's possible is that it's a vehicle for harmful chemicals. So plastic, a lot of chemicals are added during the plastic production process to give them certain properties, for example, stiffness or flexibility. And some of these chemicals have been associated with adverse health effects. For example, hormone-related cancers, autism, ADHD. And when we ingest these microplastics, these chemicals may leach out of the plastic and into our bodies. And the same is actually true for environmental contaminants like PHs, PCBs, heavy metals. These sorb onto microplastics in the environment. And when we ingest these microplastics, these may leach into our bodies. Then a different way how microplastics may affect our health is via particle toxicity. What I mean by this is the actual toxicity of the plastic. So when we ingest it, what does the plastic do to our body? But before I go into this, I'd like to speak a little bit about particle translocation. And by this, I mean the capacity of the plastic to move from the gut to other parts of the body. And this, I'm forgetting one thing, excuse me. Because in the past, it was believed that when we ingest microplastics, these are excreted again via our feces. But more and more research is showing actually that these particles can travel the gut barrier and move into the blood. And this has actually been demonstrated for nanoplastics by Valkzak and colleagues, both in vivo and in vitro. In an in vitro study, they showed that almost 8% of the particles administered traveled across the gut barrier. And when they did a similar research experiment in rats, they found that these particles travel to various organs in the rats, including the lungs, testes, spleen, kidney, and the heart. So this makes us think if the nano and microplastics can reach the bloodstream, do they also have the capacity to cross other barriers in our body? For example, the placental barrier in a pregnant woman. Well, this was investigated by Kloot and colleagues in vitro. They used an in vitro placental model. And what they found was that polycybrid nanoparticles managed to travel across this placental barrier. An in vivo study was also done by Funk and colleagues in mice. And what they did, they injected nanoplastics in pregnant mice. And within 4 hours, they could detect these plastics in various organs of the fetus of the mice, including its brain. And the fact that nanoplastics can travel across the blood-brain barrier has also been demonstrated for fish. For example, Madsen and colleagues, they showed actually that nanoparticles can be moving through the food chain. They showed that zooplankton containing nanoplastics are being consumed by a freshwater fish, and then the nanoplastics end up in the brain of the freshwater fish. And what they also saw is that these fish were exhibiting behavioral changes, and they attributed this to the exposure to the nanoplastics. And then this different study by Brun and colleagues, they exposed larval zebrafish to nanoplastics. And what they found also that they exhibited strange behavior. They dove into this and they found that the nanoplastics had disrupted glucose and cortisol levels and ultimately resulted in hyperactivity in these larvae. So these are two studies actually already showing a little bit, shedding some light on the particle toxicity of microplastics. A more thorough meta-analysis on this for fish and aquatic invertebrates was done by Foley and colleagues. And they found that overall, microplastics were capable of affecting the growth, reproduction, and in some cases, even survival of these species. And very little work has been done on the particle toxicity in humans, but this study by Sherinsky and colleagues showed actually that nanoplastics microplastics, they cause oxidative stress and ultimately cytotoxicity in human cells. And then one of the very rare studies really looking into the health effects in humans due to microplastic exposure was done by Eschenbacher and colleagues. They investigated and studied nylon flug workers. They found that they experienced a lot of coughing, breathlessness, and reduced lung capacity. They looked into this and they found that this was caused by the nylon fibers, as these cause lesions in the lung tissue. And then the third and final way how microplastics may affect our health is because they may act as a vector for human pathogens. So microbes tend to use plastic as a substrate to grow on. These microbial communities that grow in the plastics are also referred to actually by the plastosphere, in this case by Zettler and colleagues. And they point out that in this plastosphere also human pathogens may be present. And this was actually also found by Kirstein and colleagues. They found the genera Vibrio on microplastics. And some of these were, some of the species that they found were identified as human pathogens. But then it's a very important question to ask, of course, what about human exposure? Because without exposure, of course, there's no risk. Well, as early as the 90s, plastic fibers have been found in human lung tissue. And much later, in 2018, microplastics were reported for the first time in human feces. And around the same time, research started coming in about the pollution of microplastics of our indoor environments because of our synthetic curtains and synthetic carpets and furniture. And what they found was that an average household produces about 20 kilograms of dust each year. And of these 20 kilograms, 6 kilograms exist of microplastics. So it shows that we actually might be exposed to microplastics in our indoor homes quite a bit. Then this other study in 2019 came out by Cox and colleagues estimating the intake of microplastics by an average American. And they found that an average American ingests and inhales around 100,000 particles each year. However, they point out that this is probably an underestimation because they were only able to include 15% of the diets because data is lacking for most food items. So they could only assess 15%. And then in the same year, a study came out showing that plastic tea bags released billions of microplastics in our tea water. So slowly but surely, with new research coming out, we're starting to find out that our exposure to microplastics might be a lot higher than we initially presumed. And based on all these research studies on our exposure, on the particle toxicity, on the possibility that microplastics act as a vector for chemicals, for pathogens, we are concerned. We at Plastic Soup Foundation, but also the Plastic Health Coalition, we are concerned. And this is why we feel a lot more research should be done into the health effects of microplastics. So what we did last year, we organized a summit, conference, the Plastic Health Summit. And in the summit, we brought together all leading scientists in this field, innovators, industry, policymakers, to get together and talk about this issue and discuss, does plastic make us sick? And to give you an impression of our summit, I'd like to show our after movie. And like I said in the beginning, I hope the delay is not too bad.
Welcome to the first Plastic Summit. We're going to talk about how toxic it all is.
Does plastic make us sick?
Today I wanted to talk to you not about the chemicals, but the possibility of plastic particles in our blood.
I am worried about what's on the microplastics. Microbial populations may grow.
What is the consequence of those chemicals and plastic leaching into food?
I do see a light at the end of the tunnel. Redesign is the central, crucial R for solving this problem.
Many of the early warnings that have been studied by the EEA have turned out to be legitimate problems.
I believe we need a new strategy.
I think there is no time to waste here.
And I think the last thing that the world wants now is another environmental nasty surprise.
The hope is right on the horizon. With all the resources we have in this room, I have no doubt that we're going to do this.
The greatest threat to our planet is the belief that someone else is going to save it.
Human health is an incredible motivator for change. Cigarettes, lead petrol, DDT, sun cream. The human health risk of plastic poses an incredible opportunity for much greater change.
The future must be together.
If you think the economy is more important than the environment, then try holding your breath while counting your money.
What I saw this afternoon is that some things are scary, some things are reassuring.
Being out there and cleaning up, doing it on the street, doing it on the beach is the best way to reflect. Is it the solution? No.
So it's our role to all work together to that end, I think.
We are adepts of plastic because it's pushed upon us. They say we need it, but we don't want it.
Because right now, it's not anymore about convincing people that plastic is bad. It's about how do we get out of this crisis.
I think it's even essential that we take other groups aboard here. We want to have the evidence on the table as soon as possible.
The person that comes with a new material, the group that comes with new material to replace single-use plastic bottles tomorrow, they will make billions.
Thank you so much, all of you, for this panel. Thank you.
With the media coverage.
Today, I would like to tell you something about a project that I'm doing funded by SonMW.
This is not right. Yes. One moment. Yes, what I wanted to say was that with the media coverage that we achieved during this day, we managed to reach over 400 million people worldwide. So we were very pleased with this result. And what I also wanted to point out actually is that one of the speakers during this day was Dr. Susan Shaw. She was also featured in this after movie, and she's one of our very valued partners in our health coalition. She'll be speaking later this day. So what we also co-organized this summit with ZonMW. This is a organization, a Dutch organization financing health research. And in early 2019, they initiated a research program called Microplastics and Health. And in this program, 15 research projects started looking into the effects of microplastics on lungs, the digestive system, the immune system, and the potential of particles to move to other parts of our body. And 2 scientists in this research program shared some preliminary results during our summits, and they both gave excellent talks. So I would actually like to show 2 fragments, if I have the time, of both of their talks. Associate Professor Nienke Vriesekoop, she spoke about how immune cells deal with microplastics, and Dr. Francine van Dijk, she spoke about the impact of plastic microfibers on our lungs.
It really prompted us to ask the question, what now if immune cells are encountering microplastics rather than bacteria? Will they also start to engulf them? Will they degrade them, which of course they actually cannot? So how do they deal with that? Do they try to attack them but they cannot? So this is what we wanted to investigate. And in order to do this, what we did is we isolated blood cells from humans, from human volunteers. We put them in a culture dish, and then we put them together with 4 types of microplastics. So we chose to look at 2 sizes, the 1 micrometer and the 10 micrometers. The 1 micrometer is about the size of a bacteria, and the 10 micrometer is slightly smaller than a cell. But then in addition to looking at clean microplastics, we wanted to look at another situation, because normally actually microplastics are not clean. They tend to stick to many things. So we were thinking that if we would encounter microplastics in our body, it's much more likely that blood components will start sticking to these microplastics and perhaps changing how your immune cells are dealing with them. So we also thought it was important to not only look at the clean microplastics, but to also look at the microplastics that had these sticky blood components added to them. So this is what we did. And here you can see a microscopy picture. Now in gray are the immune cells, and in green are the microplastic. These are the bigger beads. They're about the same size as the cells are. And what we can see in the uncoated or the clean microplastics, that the immune cells bump into these microplastics, but they're not engulfing them like they do with bacteria. But then, if we have the microplastics that are coated with the blood components, we see a very different picture. We can see here at the bottom that, and also in some other cases, that now these microplastics are being engulfed by immune cells. But what was even more troubling is that we could see the cells dying, and that you can see in this movie with a red signal. So you can see gradually in this movie, also the cell there at the bottom that had engulfed the microplastic is now starting to die and flashing this red signal of death. So that was worrying to us. Then if we compare that to the smaller microplastics of 1 micrometer, about the size of a bacteria, we found that also they were being engulfed by the immune cells, but in this case we couldn't detect the death signal. So it seemed like really the bigger microplastics were giving a bigger impact.
So next I would like to show the talk, fragment of Francine's talk. So she looked at the effect of microfibers on lungs.
Dr. Sophie, sorry to interrupt you.
No
you
worries.
have only a few minutes.
Okay, then I will skip her.
Yeah, please. Yeah.
Yes, that's fine. I can. All videos of the summit are available on our website, plastichealthcoalition.org, in case anyone is interested.
Yeah. If you can wind up, we'll have a question-answer session followed by your talk. Yeah.
Yeah, I will try to finish soon. Yes.
Thank you.
We wanted to have a second edition of this Health Summit in October this year, but we had to postpone it due to COVID-19 to April next year. And in the meantime, we want to keep disseminating important scientific research, and we'll do this via our Plastic Health channel, which we will be launching soon. And in this channel, scientists will get the opportunity to speak about their research. I want to finish with some research that we disseminated last week. This concerned two studies, one by Professor Willy Pijnenburg and one by Professor Margherita Ferrante, a Dutch scientist and an Italian scientist. And Margherita Ferrante, she purchased vegetables and fruits from markets in Italy and examined these for the presence of microplastics, and she found alarmingly high levels of microplastics in these products, with apples and carrots being the most contaminated. And at the same time, Professor Willy Pijnenburg intended to publish a research showing how actually microplastics can move into a plant and up to the edible parts of the plant. Because in the past it was thought that microplastics were too large to penetrate healthy plant tissue. And with this, we sent out a press release, and with this dissemination, we managed to get over 55 pieces of coverage all over the world, from the US to Europe, Turkey, Taiwan, Indonesia. And together, all these media had an online readership of over 2 billion people, and an estimated 3 million people actually read about this research in the last week. So we were very pleased with these results. So therefore, I'd like to say, if you are a researcher, you're doing plastic and health related research, we'd love to get in touch with you. So if you want, please reach out to us. Thank you so much, everyone, for your attention. And if you have any questions, I'm happy to.
Dr. Sophie, thank you very much for your excellent presentation, which is now a hot topic now.
Okay.
So we have a lot of questions. Most of the questions are very in general. Okay, we can just generalize it and put it as a single question.
Sure.
Okay?
Yeah.
So one general question asked by many is, what happens when the microplastic enter into the bloodstream, and what is the fate of it?
Yeah, that's a very good question. And well, based on these animal studies, we see that these can accumulate in various organs. However, actually no research has been done on the effects of the particles in after accumulation. So it's still a very young research area. And particularly, of course, for humans, this has not been studied at all. So this is why we find it so important that more research will come out.
Okay. Okay. So one more very general question from the participant: does microplastic reach human through food storage plastic containers, which they use day to day?
Yeah, there have been studies showing that plastic food packaging can contaminate food items with microplastics, yes.
Yeah, thank you. Dr. Priyanka, do you have any questions from the participant? Yeah.
I picked up the same two questions that you asked, Dr. Prakash, and there is just one more question that Dr. Sarkar has asked. That like this is a basic question, but very relevant to the subject. What are the different types of solvents in which these microplastics are soluble?
Sorry, can you repeat the question? I didn't hear it.
The solvents in which microplastics are soluble.
The solvents?
Yes.
I'm not sure if I understand the question. I'm sorry.
Like she's asking, in which type of solvents are the microplastics soluble?
Ah, okay. Yeah, that's not something I'm not working, like I'm working as a researcher, but I'm not doing any experimental work myself. So that's a question you should ask to a particular researcher doing that kind of experimental work. I'm sorry, I can't answer that question.
No problem.
Yeah,
No problem.
it's
Thank
okay.
you.
Thank you very much, Dr. Sophie. So due to time constraints, I think it's difficult to answer all the questions now. But anyway, the participants will get the answer either a QA box or through email. For the
Okay, yeah, everybody can always reach out to me via my email, of course.
Yeah. So once again, I thank Dr. Sophie for your presence and presenting your excellent talk. Thank you. Thank you.
Thank you
Sophie.
so much for having me. Thank you.
Yeah.
Thank you, Dr.
Yeah, stop sharing, of course. Oh, all right.
Yeah.
I'll share the screen for the next speaker.
Yes.
Yes.
Okay, should I start?
Just hold on, Dr. Alba.
Okay, thank you.
Yeah. So, so the next speaker is Dr. Alba Hernandez from University of Barcelona. So she's the leader of environmental carcinogenesis section of the mutagenesis group at the Autonomous University of Barcelona. And she's an awardee for Young UAB Researcher Acosta in 2012. Okay? And she is also a member in European Environmental Mutagenic Society, Spanish Society of Environmental Mutagenesis, and Consortium of Biomedical Research in Network of Epidemiology and Public Health. So her current research focuses on the potential transforming effects of non-materials under chronic exposure settings. She has published over 50 papers and has participated in more than 20 projects related to the nanotoxicology field. So now I call upon Dr. Alba to present her talk. Yeah. Please, Dr.
Okay, thank you.
Carry on. Yeah. Can you share your slides?
Yes, are you able to see the screen now?
Yeah.
Okay, yeah?
Yes.
Yeah.
Okay, so I will start my talk that is a research work with title Polystyrene Nanoplastics May Pose a Health Risk for Humans, and it will deal with the genotoxic and secretory effects of this compound in blood cells after ex vivo exposure. So I have chosen these pictures to start with my presentation because I believe that it's important for two different reasons. The first is that I do believe that it illustrates very well that plastic pollution has become a global environmental health problem impacting the whole world. And the visible part of this problem is seen in this huge plastic island that we can find in the sea, and that is visible on the surface, as you can see here in those pictures of this Pacific garbage patch between California and Hawaii that is 3 times the size of France. So the second reason that I think these pictures illustrates very well is the fact that for many years, for decades, I would say, plastic pollution has been considered a problem circumscribed to the ocean, and with the possibility to endanger marine species and marine wildlife, but very unrelated from humans and human health. So all this misconception, I would say, has changed with the existence of microplastics and nanoplastics originated from these macroplastics that we can see here. As we have learned that these micro and nano-sized plastics are able to be eaten from different marine species and therefore entering the food chain and reaching humans. As in the previous presentation, Sophie Funk has also explained, we have also learned that microplastics and nanoplastics can be found in the drinking water, in tap water, bottled water, not only in the water from the ocean, but also in rivers. And also, we have learned also that these micro and nanoplastics can be found in the air of big cities, and even in the air of very remote, non-populated areas everywhere in the world. So polyethylene, polypropylene, and polystyrene are the most used plastics, and therefore are the plastics highly present in the food, in the water, and also in the air of the environment. So this picture here summarizes all this situation where we have the main responsible for all this, that is the plastic garbage that can originate these micro and nanoplastics that enter into the environment, which are ubiquitous in the whole environment. Of course, humans can be exposed to this micro and nanoplastic, as I've mentioned, the main polypropylene, polystyrene, and polyethylene through the water, through different food and beverages, through the gastrointestinal tract, and also by inhalation of the contaminated air. So micro and nanoplastic is now also a human problem and a health problem. This situation comes with a lot of unknown facts around the micro and nanoplastics that I have summarized some of them here. The first thing that I would like to point out is that we lack proper methods for identification and quantification of these micro nanoplastics. So it's really, so we don't have really certain information about what are the exposure levels that are able to reach humans. So there's not a clear understanding of the occurrence of micro nanoplastics in the environment. These facts, the lack of methods to really measure the particles, the plastic particles, and the unknown exposure levels also are combined with the lack of clear information about the toxicological effects of these micro nanoplastics. We have no certain information about how toxic these plastics are, and neither about how genotoxic these plastics are. This means if they are really able to come in contact with DNA in our cells and damage this DNA. There is literature regarding this toxicology and genotoxicity from the ecotoxicological point of view, but not with mammal systems. Therefore, the knowledge on this fact in humans is really little. Also, we have some evidences of transport of micro nanoplastics across the gastrointestinal and the respiratory tract. But again, this information in humans and human models is very limited. So we don't clearly understand how it accumulates in cells, how cells uptake these particles, whether they accumulate in the primary and secondary organs, and how it distributes through blood. To produce different effects, such as immune effects and long-term effects after some time of accumulation in the different organs. So all these knowledge gaps prevent from doing risk assessment or adequate risk assessment. So the truth is that at this point, the scientific community is not able really to give solid data to regulators so they can properly deal with this problem and protect the population exposed by taking action on this environmental problem. So this is where our laboratory comes and to deal with these knowledge gaps, trying to shed some light on this and get really solid data on the potential health effects, the potential toxicological effects of micro and nanoplastics, so we can get this solid data to help regulators and to prevent the population from being exposed to micro and nanoplastics. So we have been working with some models from the nanotoxicology field that we have translated to this micro nanoplastic world, let's say, or some of these models are meant to move away from using animals in research. So we have worked with a model of the gastrointestinal tract. So we are able to analyze the effects of micro nanoplastics in the gastrointestinal tract in vitro. We have also used an ex vivo blood exposure system to study the immune effects. And we have also another system to assess the long-term exposure effects of micro nanoplastics, which we believe is really important because we are all exposed to these micro nanoplastics that are everywhere for many, many years. So we have a system in vitro to to chronically expose the cells and analyze the long-term effects produced by plastics. In this talk, I'm gonna center on this ex vivo blood exposure to assess the effects of micro nanoplastics in the blood system. I would say the evidences that made us move to this system come also from an evidence from the gastrointestinal barrier. So the model that we are using to study the gastrointestinal interaction with plastics is a complex model with differentiated enterocytes combined with other cells able to secrete mucus and also immune cells, cells that are able to convert these enterocytes or differentiate these enterocytes into immune cells. So we have a complex barrier model that grows in this kind of transwells. So we can analyze, as in the pictures that you can see, let me move. Here, we can analyze these cells. This is a confocal microscopy image, and you can analyze the cells in these transwells. You have the cells here on the top, and this is a section that you can observe here, and this other section that you can observe here. So we are able to visualize the micro and nanoplastics if they are fluorescent, as is the case that you can see here, here, or here, to analyze if they can translocate through the gut, through the gastrointestinal tract. Interestingly, when we started working with nanopolystyrene, we observed that this nanopolystyrene can very easily enter into the cells in the barrier system. And even more interestingly, that this nanopolystyrene that you see here is able to cross this barrier, translocate through this barrier, go to the basolateral compartment and reach other cells that are placed, lymphocytes, immune cells that are placed in this basolateral compartment. So we demonstrated here that this nanopolystyrene is able to translocate through the gastrointestinal tract. And this gave us the hypothesis to start working with the effects of this nanopolystyrene in the blood system. So the ex vivo exposure system that we work with consists of having or drew blood from different donors. I have illustrated here one donor. The work that I'm going to show the results it's a combination of 10 different donors. So for each one of the donors, we split the sample in different parts. So we can treat the same sample as a negative control, a positive control with lipopolysaccharide, and also different doses of the nanoplastic, in this case, nanopolystyrene that we wish to analyze. So we are, after all the work, comparing the different doses or the different treatments with its own negative control. All this during this incubation with the nanoplastic, blood is in agitation at 37 degrees. So we are sure that these particles or these plastic particles doesn't go to the bottom and doesn't go in contact with the blood at all times. So with this system, we analyzed the effects of this nanopolystyrene that you can see here characterized. We work with two different compounds. One is the pristine material that you can see here, and the other one is a label with a fluorescent mark, so we can track the behavior of these particles inside the cells. And here you can see the different characterization parameters, where you can see that we have some degree of aggregation, but that in general particles are well dispersed and in the size claimed by the supplier. The toxicity up to 100 micrograms of milliliters of this nanopolystyrene, either in the pristine or the label form, is flat here. There's no toxicity. It doesn't cause toxicity to the blood cells between 24 and 72 hours. And this is not new. This is something that many researchers have found. Usually there's a consensus that plastic particles are not toxic in many of the systems analyzed. These particles are able to very easily enter inside the blood cells. Again, these are confocal microscopy images. You can see here, these cells are lymphocytes. These are polymorphonuclear cells, and you have here monocytes, and as you can see in green here, those are the nanoplastics that we have put in contact with the whole blood in agitation for 24 hours. There's some degree of accumulation in these polymorphonuclear cells. We have a lot of accumulation in monocytes and barely in lymphocytes. And this is, as you will see later, a very important point of this work. And here you can see the different populations after cell sorting, so they are separated. And as you can again see, the lymphocyte doesn't show or barely show some internalization of these particles. Polymorphonuclear cells show particles inside, and a lot of these particles inside the monocyte, which is not strange, as these monocytes are phagocytic cells in the blood system. So we have quantified and not only analyze this internalization as in the previous slide as a qualitative way. We have also quantified this internalization, and as you by flow cytometry with the gating system that you can observe here. Those are lymphocytes, those are monocytes, and those are polymorphonuclear cells. And in the negative controls, you see that there's no cells in this area here in this gate, while when you expose the blood, the whole blood to the nanoplastic, you start visualizing these cells here that represent the cells that have been able to internalize these plastics. The quantification of this is here in these graphs, in 24 hours, at 42 and at 72 hours. Lymphocytes is this flat line in here. So during all this time, these lymphocytes barely internalize these particles. Here are the polymorphonuclear cells that reach a plateau at 48 hours that doesn't change very much through time. These are the monocytes with an exponential increase up to 24 hours that also, again, doesn't change very much after the first 24 hours. So this means that the different particles are able to enter the cell very rapidly, and the situation stays steady for the following two days. So the next thing that we did once we really saw that these plastic particles can enter into the cells is to apply the Comet assay to study the genotoxic DNA damage. So this, just as a brief introduction, this Comet assay consists of a technique that isolates the different cells after having been exposed. These individualized cells are included in agarose and placed on a support to make this to stick the cells into this gel bond. Apply some treatment to leave only the nucleoids, and then apply an electrophoresis to visualize the DNA that's able to migrate through the right. So if the DNA in the cells is not damaged and there's no lesions, or that can be the DNA is not fragmented, you observe the nucleoid as it's in this scheme here and in this picture here, with barely no tail into the left, to the right. On the contrary, when you have lesions, single strand breaks, double strand breaks into the DNA induced by the treatment that you have applied to the cells, you are able to visualize, as you can see in this scheme here and in the picture here, the nucleus and also this tail indicating all the fragmented, the damaged DNA. So we can measure all the DNA that's in here and compare the treatment versus the controls to really check in a very sensitive way whether these compounds are able to produce DNA damage. So usually when the people work with blood, before applying this Comet assay, performs an isolation of white blood cells or PBMCs. There are routine methods like Ficolpac, Histopac, or RBCs lysis to really get only these white blood cells and get rid of the rest of the fractions of the blood that are not interesting to apply to the technique. So people usually work with at least a mixture of two different cells, that's lymphocytes and monocytes. In some cases, when we work with white blood cells, we work with three, because we include the polymorphonuclear cell into the equation. I'm mentioning this because it's important that when we analyze these comets, when we apply the Comet assay to this whole population of these two cell types or three cell types, what we see is something, as you can see here in this graph. This is the percentage of DNA that has migrated, so the percentage of damage in the cell, the controls, and two different treatments with the nanopolystyrene nanoplastics. And there's no effect visible.
However, Dr. Alba, sorry to interrupt.
Yes.
Can you please conclude within 2 to 3 minutes? Because we are running short of time.
I think so. 3 minutes, okay?
Yeah. Thank you, Dr. Alba.
Thank you very much.
Yeah.
Okay, so when we separate the different the cell population after cell sorting, and we apply the Comet assay after the cell sorting, so you can see here that we can isolate lymphocytes, monocytes, and polymorphonuclear cells, and then we apply the Comet assay. We observe a totally different situation. Lymphocytes present no damage. Monocytes start presenting damage at the higher concentration, and interestingly, polymorphonuclear cells that are the ones that usually we get rid of during the isolation of PBMCs to apply the Comet assay, show damage significantly at the two different doses that we analyze. So this is a very important conclusion because, as I said, regularly we don't do this cell sorting before applying the technique. So we found no effect, maybe because it's masked by the different populations of cells that we have in the sample. After observing this, we have also taken the plasma from the different donors, and we have applied an indirect soft agar. I don't have the time to explain how this technique works, but as a general explanation, we would say that when something is inside the cells, sorry, when the cells in the blood secrete factors, those factors are able to impact the growth of other cells in these plates. So the differences that we observe here and here after the treatment with 2 different doses of these nanoplastics is that the cells are changing the secretion of the cytokines after the exposure with the nanoplastics. And you can see this also quantified here. So there's something in this secretome, there's something being secreted by the cells that is different between the cells that have been treated and the cells that are not treated. So to study what are those differences and really observe what are the molecules that differently change, we have applied a proteome profiler antibody array to the different donors. And we have found the results that you can see shortly here. There's the 40% of the cytokines that were analyzed were found to be downregulated. Dysregulated. You have here only the ones that are upregulated and only at very high fold change, more than 8-fold change. Some of them are common between the 2 doses, but as you can see here, there's a dose dependence of the change that we can see. And here you have different graphs highlighting the different molecules that are involved in the immune response, in the inflammatory response, the stress response, and cell proliferation. So to finish and conclude with some important remarks, we have demonstrated here that nanopolystyrene is genotoxic after ex vivo wall exposure, that this genotoxicity is dependent on the blood cell type, and this has important implication for future studies, and that nanopolystyrene impacts the blood secretome after whole blood ex vivo exposure with results compatible with severe immune effects and inflammation. So this would be a nice topic to continue investigating in the future. And I would like to highlight the work made by the different postdoc and predoc students in our laboratory. And I will be more than happy to answer any question that you may have. Yeah. Thank you.
Yeah, thank you, Dr. Alba Hernandez. It was a highly informative talk.
Thank you.
Thank you very much for sharing your research work. Very interesting. Now we'll just open the question and answer session. Dr. Priyanka, can you just shoot the questions to the speaker?
Yeah. Thank you, Dr. Alba. The talk was very interesting and the experiments were very interesting. So I'll just take a few questions. There are many, but as decided, all the questions will be sent to you either by email or other means.
Sure.
I have just taken 2-3 questions. So first question is like when this nanopolystyrene is internalized in WBCs, leukocytes, are there any external symptoms? Like how does it manifest the internalization of this nanoplastic in leukocytes?
And no, actually, you have seen the confocal microscopy indicates that the cells do maintain the shape and the status. There's no toxicity, so apparently there's no damage to the cells. But as you can see, the manifestations are more molecular in the molecular side. They secrete different factors, so they respond to the treatment by secreting molecules to communicate with other cells and changing this communication, which is very important for the immune system. So it has an impact on there. And also it appears that there's genotoxicity, but external or visible other signs we have not found.
Yeah, Dr. Priyanka, any more questions?
Just one more question was there. Is there any specific reason for like a specific, some type of leukocytes taking up more plastics than the other type of leukocytes? Like in one of your experiments you have shown somewhere. So.
Well, actually, this is something that you will listen into another talk by one person from our lab. Yes, there are, but we have worked that in vitro and not with ex vivo exposure. It's an important and interesting question. Huh? So the different types of different cells, I mean, there are different lymphocytes and they don't internalize and they don't show the same damage. And there are different monocytes and they don't show the same damage. So it's really cell-dependent. And this is something that the scientific community needs to learn about all this problematic. So if we want to really understand the toxicity, the genotoxicity of these materials into the cells, we need to very carefully pick the system where we analyze this.
Right. Thank you so much, Dr. Alba.
Thank you to you.
Yeah, Dr. Alba, thank you very much for your presence. Okay.
Thank you.
Thank you. Now we will move on to the next session by Dr. Anthony Marconi. Dr. Priyanka, can you just share the slides?
Yeah, Dr. Alba, can you stop sharing the...
Yeah, please.
Dr. Priyanka, you can proceed. You can proceed.
Okay. Dr. Jayaprakash.
Yeah, next. Yeah. So the next speaker is Dr. Anthony Marconi. So he obtained his PhD in biochemistry and cellular biology in 2003, and he's working for 2 years as a clinical oncologist before joining Vigicell, a biotechnology company that became Tame Water in 2005. And Dr. Anthony strives to apply his skills in all these developments and has happily participated in the evolutions leading to this Tame Water. Yeah. Thank you. Dr. Anthony Marconi.
Yes.
Yeah, can you please start your presentation? Please share your slides if you have.
I'm in the process, yes. Thank you very much for this introduction, and I'm very, very honored to be speaking after such eloquent speakers and among many others to come. As you can tell, also, it's very special for me because I really love Tamil Nadu, and this is a special background in Chennai, which is very dear to my heart.
Yeah, thank you.
So, of course, today the main topic is microplastics, obviously. And I will take this occasion to go over, some topics that were previously addressed. So maybe I will say things that were already said, but it's also going to be the occasion for a different way of lighting those topics, I would say. More importantly, I want to focus on the challenges of microplastics presence, their impacts. Now you have a better idea of how it goes, but we can also address some other points. And one of them is the industrial context, because a lot of research is done in the laboratories, but when you try to bring the research and also try to apply it to real world commercial or not even efforts on an industrial scale, you have a set of challenges of your own. So this is what I'm going to address. And first, I'm going to tell you a little bit about what we do as a company, because we are not a public research, although we come from public research historically, we're a private company. And my computer is, okay. So I'm from the company Tame Water, and what we do mainly is we design and we market innovative solutions for water quality evaluation, right? And we mainly do this by biological methods. So we used what we call bioassays or effect-based methods to test the quality of water. Ironically, today what I'm going to show you about microplastic involves very little biology in terms of measurement.
ये भाई तो तमिलनाडु बैकग्राउंड लगाया है.
Yeah, nice, huh?
Hello.
So back in the days, you know, the kings and emperors would have gone to.
Voice नहीं आ रहा क्या?
It's Chennai Ramakrishna Mission. so you know, back in the days, emperors and kings would have people to test their water and see, I mean, or the food to see if they die. Of course, this is not really ethical. So among the years, we have developed different assays. Up to now, we have toximeters, little machines that can give a biological answer to if the water is toxic. This is our approach at Tame Water. We use mainly biology because it's a way of addressing the problem of water contamination, pollution, that physical-chemistry classical measurements cannot address, not because they are inferior, but because they're not designed to do this. So for example, you know, like health concerns and the effects of all the contaminants on the health and functions of the living organisms can only be addressed by a biological method, right? So we have services, we have products. Services is in our labs, and products is with the toximeters. That's how they look like, you know. We have several of them. We have them for drinking water, for industrial effluents. We have even one for biodegradation, which is very nice. And we do a lot of biological methods in our labs. And so we have a lot of different packs that target the main functions, the main problems, the main avenues of toxicities. One of them is the microplastics. So speaking about microplastics, let's get down to it. So as you could see from all the topics previously, and mainly also Dr. Palani Sami, plastics, it's not going anywhere. But, so we're gonna use plastics more and more and more, but it's going everywhere because we're using and creating plastics more and more. So this, you see, there are, you know, rather old data, 2015, 2017, but you can already see how much we produce, and is going to increase. And of course, because it increases, we're going to find it more and more and more in the environment. For example, these projections from the World Economic Forum in 2016, they evaluate that by 2050, by weight, you would have as much plastic in the ocean as there are fishes in terms of weight. So it's a bit concerning. We also hear about renewable energies and the end of oil and everything, but oil is needed for plastic, and the plastic share of global oil consumption is only rising. And of course, the carbon budget also. So it's really something we have to address, micro or even macro, like plastics we all know, we are all familiar with our appliances. We have to address it because it's not something that is going to disappear. So what are microplastics? So we all know what plastics are, as I said. Microplastics, you still have a little bit of fuzziness in the literature because you find studies on the sub-millimeter scale. You have studies which go all the way to 10 millimeter scale, which is like, even if you're aging like me, it's not really a microparticle because you pretty much can see it with your eyes. Or even centimeters, because when you have fibers, they are very thin, so they can be long, but they have a very, very thin diameter, so they can, some authors count them as microplastics. There is quite an accepted consensus since 2008, which is any particle of plastic inferior to 5 millimeters. But the problem is that these plastics, they are not just on their own in the environment. They can agglomerate with the natural fibers, like wood fibers, plants, suspended matters, everything. They can agglomerate with other man-made particles, glass, other polymers, which are not considered as plastic, but they can still cling to each other and make new particles. And as you saw from the previous talks also, they can combine with pollutants and become like carriers of pollutants at the molecular level. So it's quite complicated actually when we talk about microplastic. It's not just a matter of size, it's very complex. So my topic was addressing concerns from the plants to the plates. it was a play on word. I was talking about the industrial plants. Although if you're like me, you also like to have a lot of plants in your plates. So you know by now that we fabricate these plastics for our everyday purposes, and we literally cannot live without them. Literally, like if you were to remove everything plastic from you, your life would be pretty like in the Stone Age. But how do they end up in the environment? You see, there's still a lot of things that are not clear. How are they generated? How do they end up in living organisms? You saw that there were many pathways. And of course, because of their nature and because of how they can end up in organisms, what are the concerns regarding health? What are the impacts on individuals and on populations? And what when they mix with all the other contaminants that we know and that are already proven problematic. So we need to measure this if we want to tackle this problem, but it's not so easy. There are problems, there are challenges with identification, quantification, and what about the effects? So you can see this very complex question, which I'm putting in the title here. We've known that they've been described like millimetric particles that you can see with your eyes since the 70s. The term microplastic is from 2004. But even now, 16 years later, hazard, we know there's a hazard, that's a certainty. But what we're uncertain about is what kind of hazard there is, like, because it can be dependent on the size of the particle, as we saw on the nature, which means that the polymers and the various plastifying compound added. Also, organisms may not be sensitive. A mouse and a rat and a human and a mussel and a fish might not be sensitive in the same fashion, regardless of the exposure mode. And are these effects maybe, okay, just little, so it's not so concerning, or is it really a problem for some population? Again, it's multifaceted. You could see that you can eat them, ingest them, you can breathe them. Who knows? You know, some organisms also can internalize them by contact. How do they transfer to our plates? What if you eat a lot of fish and your neighbor eats only plants or only meat? These people will have different exposures also. So you see, it's again another layer of complications. Okay, I stole this diagram from Zhang in one article. It's a bit busy, a bit complicated, but that's the point. I mean, if you don't look at the details, you can see that the contamination continuum is very complex. We create it, it gets disseminated, it gets accumulated somewhere, and then it comes back to the cycle in another form. It's really mind-blowing. So you can see you have like obvious activities of plastic creation, but you also have less obvious one, like you saw from Professor Sewell in the beginning. Like, if no one told you, would you know that there are microplastics in personal care products? If no one told you, would you know that your laundry machine does produce microplastic? Because the things we wear, I think this shirt should be natural, but who knows, has synthetic fibers. So of course, a lot of the plastic you find in the ocean comes from terrestrial sources, rivers being the dominant pathway, and also they mostly come from poor management of waste. So what are the effects? Well, the first effect for a researcher is headache, because how to tackle this problem? Again, you look at the literature, you have a lot of different effects described for individuals and populations. For like living sea-based, water-based organism, you can find loss of appetite because you eat little particles, and in the end, in your system, it makes you less hungry for everything. So it can devolve into a loss of energy, change of behavior, the development, the growth can be affected. So if you affect this on an individual level, you can also have an impact on populations. What is the toxicity of these particles? Again, you cannot have a clear-cut answer of one mode of toxicity, which we call adverse outcome pathways. So the literature described like tissue necrosis, which means cellular death at the tissue level, oxidative stress, the immune response, neurotoxicity, genotoxicity we just saw, some other subcellular problems with the cell machinery. Again, different organisms might not be sensitive in the same fashion. Like for example, a mouse and a rat for most of the people, it's the same thing, like very similar animal, but have an immune system that is very different. So one might respond very differently to the other. And there are other adverse outcome pathways that sometimes we are surprised to see, like what does it do to the gut microbiome? Even if it doesn't do something to you as an organism, if it does something to the bacteria that have a good relationship with your digestion and nutrient intake, what does it do? So we see a lot of headaches, but there's more headaches for the research. Again, those size, tight dependence, what does it do when it combines with other pollutants? You can see a diagram on the right here, because there are many ways of combining, releasing, and all of this phenomena. Also, microplastics can become like leachers for the contaminants that are attached to them, and even vectors for invasive species, something also we don't think about much. There was a very nice paper that showed that that found terrestrial bacteria, but in a marine environment, and they adapted to the marine environment because they were stuck to microplastic particles. It's really, really a complex interdependent issue. And last set of headaches, industry headaches, because bringing a method that works in a laboratory to a market when you need to do a lot of assessment, when you need to make money out of it because you're a private entity, it's challenging. You know, in terms of innovation, we speak about the valley of death, which is like, you see a lot of methods in the lab, you see the industrial applications, but it's very hard to go from one to the other. This is the famous valley of death. It can be a problem of scalability. What you can do with a student in the lab on the bench, one experiment a day, when you need to make 24 every day with standardized methods, it's not so easy. It might require adaptations. It can be also a problem of expertise, like I need this very complicated machine to do the measurements. How do I do it? Well, I need to buy the machine. I need to have highly trained people operating it, and maybe it's not commercially feasible. Or it can be a mix of all of this. But in spite of this, industrial developments are really needed, again, because it's a global problem. It's a problem that is going to get worse. So we need industries which can take the problem on a larger scale to be involved on this matter if we want to assess things better in terms of exposure, presence, health effects, everything, and information to the population, all the stakeholders. Because there's no two ways about it. We have to face it, and it's going to get worse if we don't. So that's what I summarize in this slide here. Academic laboratories may not be sufficient in number, or sometimes they're not willing. You know, we come from public research, and a lot of time we collaborate with public research. And once we collaborate up to a certain point, they say, you know, we like you guys, but it's not our job to do this, or we don't want to do this because we need to be working on something else. And so someone has to take those methods. So it's up to the private entities a lot of the time. But everyone needs measurements. The industrial that may release plastic and may be considered as pollution generators by the general population. The resource management, the urban planners, the consumer associations, everyone needs measurements, because you cannot treat a problem that you don't measure and quantify appropriately. So that's why, okay, I'm talking about the French example because I'm French, but the world over, the biggest water producer, food producer, personal care, they're already very involved. They're not, and contrary to public perception, they're not always trying to hide things or like, no, don't do this because of lobbies. Some of them are really concerned because it's vital for their business model. They know that they cannot hide the dust under the rug. So it's in their interest to like act positively about it. A telling thing is you go to Thermo Fisher and Horiba, for example, but there are many others, companies which are involved in making detectors and labware, laboratory apparatus. Now they have specialized products and even whole sections of their website that talk about microplastics. So it means that, yeah, definitely something is happening. So what do we do to tackle microplastics? We use the Raman spectroscopy, right? Because it has good spatial resolution when you stay above the micromillimeter scale. It needs expertise. That's downside, and it's time intensive. But we do have the resources, and we also have the resources for spectral database elaboration, characterization, all of this. We have a long-standing experience in the spectroscopy. We have PhDs. We worked on complex biological imprints. So it was only natural for us to extend that expertise. And we also have access to very, very cool devices. So we have an analytical workflow that enables us to identify and quantify. For the quantification, we teamed up with Suez, which is a leading global, one of the world's largest producer of water treatment solutions and you know, expertise, drinking water, wastewater treatment. They do everything. So we have those spectra that we acquire with Raman. We know how to clean them. We know how to standardize them, which is very important. I won't get into the detail now, but you can ask questions later. I don't have time, and it's very complicated anyway. It's not my specialty.
Yeah, Dr. Mark, sorry to interrupt you. Can you just wind up in 2 to 3 minutes?
I have my little timer. I'm on time.
Yeah, thank you. Yeah, sorry for the interruption.
You're welcome. So this is a slide that my colleagues and collaborators at Suez given us to highlight their sampling and analysis protocol for quantification, which has been validated with a ring test of laboratories, which is very important. And they include a lot of blanks, which is also something that is very overlooked. That's what I highlighted it in the slide. So it's a very, very standardized process, very nice. See with the interlaboratory test. So it gives us like very complex diaphragm with cartography and correlation maps. So we can establish heat maps and everything to measure appropriately. And I will conclude now. After all this talk, you know, okay, Dr. Marconi, what did you find? Unfortunately, I cannot really talk about it because it's quite confidential. It's quite sensitive for a lot of our clients. If you know my Tamil audience will know the man on the right, and you don't mess with the man on the right, right? What we can say, this has been said, and I will end my talk on this. Of course, conventional wastewater treatment reduces microplastics when you see what's coming in and what's going out. But it's all, most of it is transferred to sludge. So it's only displacing the problem. What we do with the sludges, as Dr. Palanisamy highlighted, complicated. And plastic is everywhere. So even the equipment, the measurement, the things that are designed to clean the microplastics may emit microplastic, which is ironical. And also what we learned, and I will end my talk for real on this, is that public perception is quick to point the industry or like, oh, they are polluters, they are polluters. But since it's not so easy, when you look at the water itself before the industry, you can see that the water itself is not really clean. So the industry might not be the big culprit. And so, Nandri, and thank you everyone for your attention. I will be delighted to answer a question. And you have emails of my colleagues also here that you can address for those, any kind of question you have. Thank you very much.
Yeah, thank you, Dr. Anthony. I think we have a few minutes for a question answer session. Dr. Priyanka, can you please, yeah.
Thank you, Dr. Anthony. It's a very interesting, informative talk. I just have one question. Like there is a lot of confusion surrounding this microwave safe plastic containers. So I want to ask, are they really safe? Like it is said that if you heat up the food reheat the food in this plastic containers, there is a chance of plastic leaching into the food. Is it true, or they are really safe?
Well, the scientist in me will say that nothing is safe if you push it too far. So they might be safe in the conditions that they were tested, which means a given protocol. They might not be safe if you keep the containers 5 times the time that it was tested, or if your microwave is very powerful, or if you eat acidic food, because acid can leach faster. So if it's in the container, there's always a chance that it will end up in the food. Microplastics, I mean, safe means like we couldn't detect things when we tried in these conditions. But what about the other conditions?
Yeah, so one more general question from the participants, which I have found. So participants are asking, what about the biomagnification in human beings?
Hey, well, first, it's not really my specialty, and again, I'm going to refer to the literature and also the previous speakers. It is definitely a concern. It is definitely a concern because we know some organs of ours are better at accumulating, lipo fat soluble compounds. Some organs are clearance organ with little, very tiny tubes that could get clogged. So definitely, I cannot give you like a specific answer, but liver, kidney, fat tissue as well. When very obese people lose a lot of fat, it's been shown that they build up a lot of toxicity in their body because a lot of pesticides and everything stored in the fat. And so it could be the same for the plastic particles, lungs, of course, all these things. And just to take 2 seconds also on that topic, it can skew a little bit the research because, for example, if you look at the research in mice, you see a lot of liver or kidney toxicity. But because these organs obviously might accumulate more than the others, and because also if they accumulate more than the others, you tend to focus on it for your article, but who knows about the other organs?
Yeah, I think we'll wind up with one more general question. Can you comment on microplastics as aerosols? Do you have any?
Well, yes, it's been shown that you can inhale them and that they exist in the atmosphere. Yeah, definitely. There might be smaller kinds of microplastics than the one we find in ocean research for obvious reason, because you don't see 5 millimeter particles flying around. But you have also the nanoplastics, which is a concern, but the nanoscale makes things even harder. And definitely we inhale some. Fibers, microscopic fibers, dust, everything, they're released from your clothing, from your environment. You breathe them, it's a certainty. What is the exposure and everything, then that's where the uncertainty lies. But the fact that it happens, it's true.
Yeah. Dr. Anthony, thank you very much. We are honored by your presence. Now, on behalf of the So on behalf of the conference organizer, I thank all the speakers who gave the talk. Excellent. So now I'll move on to Dr. Priyanka to take over.
Thank you, Dr. Anthony, and I thank all the speakers from this session. The talks were really informative, very interesting. So thanks for being the part of this conference. I thank you on behalf of all the hosts, the conference organizers. Thank you very much.
Thank you.
Dear participants, we will have the next session at 2 p.m. 2 p.m. post lunch. So please be there. Thank you. Thank you very much.
Bye.
Thank you.
Bye, Anthony.
Bye.
Bye. Thank you very much.
Bye, Anthony.